everyone, welcome to GDC classes. So we are back with the 11th video for our 100-day biopharmaceutics challenge in which we are going to cover the kinetics of protein binding, right? So in the protein binding by far we have completed what exactly it means, how it affects the drug distribution and what possibly can our proteins be for the binding of the drugs and how do we differentiate it with the tissue binding. So let's see what are our different proteins first and then let's move to the kinetics of the protein binding. So in the previous video as well, I have mentioned the names of few plasma proteins to which our drugs can actually bind. So number one, we have albumin. So it is most evident that most of the drugs actually bind to your human serum albumin. This human serum albumin is actually having four different sites to which the drugs can bind. First site we call it as warfarin and azapropason binding site. Second we have is the diazepam binding site. Third we have is the digitoxin binding site. And fourth we have is the tamoxifen binding site. So generally the acidic drugs, please note down what I say. Generally the acidic drugs try to more accurately bind to the human serum albumin which weighs around 65,000 Dalton. These are some important informations that you should know about, right? So these are actually the four different binding sites to which our drugs can actually bind, right, with a molecular weight of 65,000. Then we have our globulins in which we have alpha 1, alpha 2, beta 2 and GABA globulin as well. So very important, you can spot their other names and to what drugs it can bind. So we have, other than this, we have alpha 1 acid glycoprotein. Please note, we have alpha 1 acid glycoprotein which and we have lipoproteins. Right, this alpha 1 acid glycoprotein actually binds to the basic drugs, not acidic drugs. Human serum albumin binds to the acidic drugs. The drugs that may bind to globulins will indicate alpha 1 globulin we have. In the alpha 1 globulin we call it as transcortin, very important. So in its name only we have core. Core means the example will indicate steroids, mainly your corticosteroids. So it will be your corticosteroid binding site, right? In the alpha 2 globulin, we call it as ceruloplasmin. So ceruloplasmin mainly binds to your vitamins and cupric ions, right? We have C, we have U. So we can just try to correlate that yet copper is there. We have vitamin water soluble ADEK that binds to the alpha 2 globulin. Then we have beta 1 globulin which we refer to as transferrin. In the name we have ferrin which means it binds to none other than your ferrous ions. We have beta 2 globulin as well which is binds to the carotenoids and gamma globulin which binds to specifically your antigens, right? So I think now you are clear with what are different plasma proteins to which our drugs can actually bind based on their nature of the drugs as well. For example, acidic drugs are more susceptible that they can bind to the albumin and basic drugs more to the alpha 1 acid glycoproteins. These proteins differ based on what kind of drugs they can bind to as well as their molecular weights as well. Okay. So now which was our main topic, that means the kinetics of protein binding. So the kinetics of protein binding is very simple, right? We know the protein and drug actually bind with each other in a reversible fashion. That means the complex will be formed and this can be competitive, the displacement can be possible as well as reversible, right? So protein, free protein and drug is binding to form a protein drug complex. For your rate constant, this is your association rate constant. So it will indicate the concentration of product which will be none other than your concentration of protein drug complex upon your free concentration of protein and the concentration of your free drug, right? So from this equation you can get a simple equation for the your concentration of drug which is protein bound, that will be association constant into protein concentration into drug concentration, which is what is mentioned here. We have to use this equation in the upcoming correlation. So I think first thing is clear. P stands for the protein which binds to your free drug, forms the protein drug complex, forming the association constant which indicates the product concentration with the reactant concentration. So the product concentration, that is the protein drug complex, we have is the Ka into P into D, right? Moving on, you got the concentration of the protein drug complex. I'll write it here for you all so that you again remember, because we will use it. So for the you have protein drug complex, right? It will be Ka into protein concentration into drug concentration. This is from the previous equation. Now, if you had to calculate the total protein concentration, that means the protein that is still freely available as well as the bound fraction that is available, right? So this is the total protein concentration, stating that how much protein has been bound to the drug plus your free protein that is available. Okay, so if R is actually the number of moles of drug bound to the total moles of protein. If you had to calculate R, which is the total moles of your drug which is bound to the total moles of protein. So it is safe to say that R will be PD upon P. This PD is in none other than your protein concentration which is in the bound form with the drug and the total protein concentration. We already know the value of PD. We can take it as Ka into P into D. From here only we have started, right? So total protein concentration will be PD plus P. So this R value will be very much important for us. So we can have a lot of plots that we can try for the protein binding, which can include the free concentration of drug, protein, and the number of binding sites as well. So we have Ka into P into D. In the bottom part, we already have PD, which is again can be substituted from the upper equation. It will be Ka into P into D. We have Ka into P into D plus free protein concentration. So we have just done that, you can see, and by substituting and using basic mathematics, you can get the value of R as Ka into D upon Ka into D plus 1. So this is the simplest equation by which you can get the kinetics of the protein binding. Using these equations, we can have a lot of different things. It is very important to get these plots. So there are four different ways by which you can calculate your Ka. Association constant includes the number of binding site, and as well you have a direct plot, you have a Scatchard plot, you have a Clotes plot, and you have a Hitchcock plot. Now we have a Sandberg plot also. So these plots actually differ in some sort of what are their main consideration. Are they talking about the free drug concentration, or are they talking about the number of binding sites? So that is a separate topic to watch. I think you are clear with the what exactly kinetics of protein binding, how it is, how does the equation comes in play, and what kind of plots we have, so that we can better estimate the value of R with the number of binding sites and free protein concentration. Right? Thank you so much. We will meet in the next video.